In some applications, this is the only thing we do, right? So we just say, okay, cut the DNA there, and then the rest, that's the cell. Not all the DNA in your plant has a function. Some of it is what we call junk DNA. They have CRISPRed DNA in every cell of their body.
We have a sponsor for this episode of Apple Finch Pudding. Now, before I delve into this, I want to be very clear about what we use the sponsorship for. We use it to support the podcast. This means buying subscriptions to the software we need to create these episodes or put our episodes online. This might also include some editing or some other material or software we need to create this podcast. That being said, our sponsor for this episode is SciSummary. As scientists, we need to read a lot of papers, and more papers are being published every day. As a result, reading papers is becoming more or less a full-time job for scientists, leaving us virtually with no time to do our actual jobs, which is research. This is where SciSummary comes in. SciSummary provides you with an AI-based summary of the papers you provide. But SciSummary is much more than just a summarizing tool. It lets you effectively chat with your papers. You can ask questions like what methods are being used, and it even provides you with some insights and opportunities for future research based on the papers you provide. If you want to try out SciSummary for yourself, there is a link in the description below, which will also give you a 7-day free trial. Now, let's go back to the show. Welcome to Apple Finch Pudding, your gateway into the world of science. Today's scientist is Evelien Waegneer, a postdoctoral researcher in plant sciences at ILVO. Welcome, Evelien.
Thanks. Thanks for inviting me. Yeah.
My first question is always the same. Do you have a fun science fact for our listeners?
I've been thinking, and I actually have two, but I don't know which one is the most appropriate one. So I'm going to give a small pitch for both, and then you can pick which one I share. So it's either a slightly inappropriate one about elephants, or
Okay.
In the theme of the Olympics, a sports science fact.
Can I pick both?
Okay. So this is something that I read a few years ago that I thought was pretty amazing. It's about elephant genitalia, that apparently their penis is multifunctional. They can move it around instead of just the one move that most penises can. So they can use it as a fifth leg, or to scratch their belly, or to swat flies away.
Are you serious?
I'm being serious, yes. You can find pictures online, but I suggest to do that
In a safe space.
in a safe space, in incognito mode, because maybe they're going to chase you.
Okay.
But yes.
That's a fun fact. It's unexpected, at least. Okay.
But it's, I think, one of my favorite animal science facts that I know. So
Okay. If you're up to it, could you also share your fun fact about the Olympics?
it's not about the Olympics. It's more about a specific sport, because nowadays sports you need a couple compounds to be successful. You have to have a good athlete, and also a lot of dedication for your sport. But nowadays it's also a lot about science. And for example, yesterday the Belgians, they won the time trial cycling, and a lot about that is they do science into the aerodynamics of the cyclist. And it's more about the car that follows the cyclist. I mean, anyone who has seen a cycling race knows that those cars behind them, they are like prepared for the worst. You know, they have like 15 bikes stacked on top of them. And you think, what has to happen in order to need that many bikes? Well, the thing is that, especially in time trial, if the car is very high and they follow quite close by, they push the air forward, making the cyclist faster. So it gives an advantage. But when I was watching the Olympics yesterday, and that's why I remembered, I saw that they must have put in a rule or something, because they only had 2 bikes on top, so that they couldn't use that advantage.
It's a bit cheating, actually. But
well, if everybody does it.
Yeah, well, it's a smart way, or a smart use of science.
Yes, let's just put it like that.
Yeah. Well, today we're going to talk about CRISPR. You'll probably try to dissuade me, but to me, you're the CRISPR expert. But before we dive into that, we're going to start more general. So CRISPR is a form of gene editing. Can you explain to the listeners what is gene editing?
gene editing is actually a way to change the DNA of organisms. So to start with the DNA, it's, if you have to visualize what that looks like, it's an incredibly long string. And within that string, you have information that helps the cell, how to be a cell. It has all the information to function as a cell, and depending on which organism it is, that information is going to differ slightly. And the way that information is put in that long string is actually via a specific sequence of the letters A, T, G, and C. And what gene editing does is actually, you can go to a very specific spot in the DNA or a gene, for example, and change the information in that part of the DNA. So that's in very broad terms what it does. It usually consists of two components. A part of the system that does it is going to be able to find a specific part of the DNA, and then the other part is going to be able to cut the string. And because the cell, of course, doesn't like that the string is cut, it's going to try to repair it. And it's during that repair mechanisms that changes in the information of the DNA will occur.
So we have the DNA, and actually, basically, we have a component that searches a specific spot in the DNA, and another component that is making a cut.
In that specific spot, yes.
Yeah. And why do we do that? Why do we want that cut? And what are the different ways we can do that, actually?
Well, first we want that because if you change the DNA in a specific spot where we know that is linked to a specific trait, we can change certain traits of organisms and do that in a very directed and specific manner. You have different types of gene editing, but I would say the CRISPR system is by far the most used one because it's very easy and cheap.
And so what is the big advantage of CRISPR over those other techniques? So because it's cheap and easy, or are there other things as well?
But it's cheap and easy because it's easily to customize. So I said, like, you need there's a part of it that finds the DNA the part of the DNA that you want, and with CRISPR it's so you have the GPS system that's going to find the DNA. To change your target, that's super easy. So you can quite easily say, like, instead of this, I want that. And with the other techniques, a lot more work goes into changing your target. And that's why why it won out of the other techniques, because you can change your target pretty well, which makes it easy to use. And it's also quite fast and it's quite efficient.
And why is it so revolutionary compared to the other techniques?
Yeah, I think it's quite revolutionary. Yeah, because it's so easy, so cheap, and you can adapt it to different species. So it's also been something that's been shown that it works. As far as I know, it works in every species where it's been tried. So that is quite a feat for a tool like this.
Yeah. How was it discovered, actually, CRISPR?
It's actually the adaptive immune system of bacteria, because I mean, some people don't think about this, but bacteria, they also get attacked by viruses. And one of the ways they protect themselves against that is by saving a specific part of the virus in their DNA. And then the next time the virus comes along, they know that the virus is there, and they will just cut the DNA of the virus, and it cannot, it becomes harmless that way. So, and then I think it was Jennifer Doudna that made the paper about this and said, okay, perhaps we can adapt the system to use for gene editing, and instead of targeting the virus DNA, we can maybe switch it out for whatever we want, and we have a gene editor, and that's proved to be correct. They adapted it for that, and look at us now.
Okay, and so CRISPR is then actually more or less the scissors that is cutting the DNA?
Exactly, yes. It has a GPS system that finds it, and then it has an enzyme that can just cut the DNA in two. And that's how it works.
So we know we cut DNA with gene editing, but I'm assuming that's not the end of the story. Otherwise, we just have cut DNA and that's it. So what are we doing? What are we trying to do?
Well, actually, as a scientist, in some applications, this is the only thing we do, right? So we just say, okay, cut the DNA there, and then the rest, that's the cell. The cell is gonna try to fix the cut, and in fixing this, it doesn't work perfectly, so it creates mutations. And so you have two types. You can either say, okay, I want this to happen randomly, and that's, for example, what I've been doing most of the time. I don't really mind what type of mutations happen. But then what you can also do is give a template along and says, if you're fixing it, fix it in this specific way. And in that case, yeah, you can really have a specific type of mutation.
Let's start with you use random cuts, but why would you want random mutations?
But the cut is not random. It's just that the type of mutation that happens is random.
Yeah, okay.
But it's not that I want this, but it's just that I work in plant research. So out of that random cut, I generate like 200 plants. And then I have different mutation profiles. And I just pick the one that I like the most, or that I think is most useful. But I let the plant decide itself which type of mutations it makes.
So basically you're cutting the DNA, but it's always at the same spot because of that location system. And you get a lot of random actually repair systems, the repair system is not random, but the repair is more or less random. Get a lot of different plants, you all grow them, and then you see, okay, this one I like, for example, because it has nicer flowers, or this one has nicer leaves, or whatever you're doing, if it's, yeah.
It's not exactly like this, because when you do CRISPR, you tend to work on a trait.
And can you give an example of a trait that you could be wanting to adapt with CRISPR? Are we talking about flower color or leaf size?
Yeah.
Or are we talking more about different proteins that are being created?
But all those things, the answer is yes, can all happen. It's quite
Okay.
That's why the question is hard, is because it's very broad. You can really just edit anything. But I will use the example of flower color, because that's, I think, probably the easiest to explain. So let's say that there is a gene that is going to make the color of the flower blue. Then if you go to that, you set your CRISPR, okay, go to the gene that makes the flower blue, you cut it there. And then if you're going to select for mutations that highly disturb that gene, the blue will not be made anymore, and you will get a white flower. But if you, for example, take a mutation that only disturbs it a little bit, high chances your flower is still going to be as blue as before. So then that mutation is less interesting. So you're going to pick the one, in that case, that has the biggest effect on the gene in terms of disturbance.
Another way you could use CRISPR is you add a template as well of how you want the DNA to be repaired. How does that work exactly?
It's the same system. You create a cut, but you give a piece of DNA along that's actually very or completely similar to the surrounding DNA of where you cut. And then the idea is that your repair system is going to use that DNA as an example of how it should be repaired. The problem with this is that the random system is a lot more efficient than having the template. So what you usually get is you get random repairs, and then the repairs where they use the template. So that's why in plant research, for example, it's less used, because it's quite hard to force the repair system into using the template.
That was one thing that I was wondering. So if you add some string of DNA and it has some similarities, why would the plant try to copy that?
It's because the plant already has the system to use similar DNA to repair. It's because usually organisms, they have two sets of DNA, because you get one from the mother and one from the father. And if a mistake happens in one of the two copies, they can use the other copy to see how it was before. And it's kind of that system that they use. But the problem with that system is that it's only active during certain phases of the cell cycle. So it's not perpetually active. And that's why it's also less efficient, I would say.
So a lot of people talk about CRISPR, but sometimes people also talk about CRISPR-Cas9. What is the difference there?
CRISPR is kind of the umbrella term for all the CRISPR-based gene editors. But the Cas9 is the cutter, is the enzyme that's going to cut the DNA. It's a specific type of CRISPR system, because you also have other enzymes that can cut the DNA, but there will be different conditions linked to that, and they will also maybe cut in a different way. So depending on your biological question, you use CRISPR-Cas9, so that enzyme, but you can also, for example, use CRISPR-Cas12a, and that's another enzyme. So it's a way to turning your tool into a toolbox. The first thing that was really used was Cas9, so that's the most used right now. But what I at least see the last few years, is that there's more enzymes being added to the mix that you can really pick and decide depending on what you want.
So the Cas and then a number is actually the actual scissor in a specific case or something.
Yeah, the Cas stands for CRISPR associated. So
okay,
that's always going to be there, and then they're going to add a specific number. It's also because, as you asked earlier about the bacteria, the Cas9 comes from a specific bacteria, but then, for example, the Cas12 comes from another bacteria, and that's why they all cut in a different way.
So you use CRISPR, but how accurate is CRISPR? What are some of the dangers or the potential benefits?
I would say the CRISPR system is quite accurate. But yeah, the GPS, there are some issues with them that you just have to be aware of as a scientist, essentially, because it's going to recognize a specific part of the DNA. It's 20 bases, so it's 20 of those AGCTs. But the problem that sometimes occurs is that in the whole DNA of your organism, that those 20 bases, they can occur in multiple places. And then your CRISPR is going to cut all of them. And then also the issue that can be is that sometimes when it's very, very similar, it can also cut potentially with less efficiency, but it's possible. I wouldn't say that that per se is a danger. It's just something to be aware of when you're designing your experiment. It's also something when I design my CRISPR experiment is that I check those things, like which spots in the DNA of my plant are potential off-target, what we call off-target sites, that they also can cut, but that I did not want to cut. And then I can, afterwards, when I've done my experiment, I can then check, okay, did some editing occur there, yes or no? And then again, if I see that editing occurred there, I throw them out, and and that's it.
In classical breeding, they use different techniques. Sometimes they use like high doses of UV light and stuff, but that's then more random. So is CRISPR then safer or less safe, or is it similar?
I would say you know better what happened, because as you say with UV, it's very random where you're going to get your mutations. And you can then indeed check, okay, I want a mutation in that specific spot, and with UV that happened, but then you don't know, maybe there's a lot of other places where also mutation happened that you're not aware of. Whether that is dangerous or not, well, for that you check, of course, your plants, whether they are doing something that they're not supposed to be doing. But with CRISPR, because you know so well, okay, these are all the potential sites, you at least have a very good idea of what happened in terms of mutations, and you know better than those techniques where you get random mutations, such as with chemicals or with UV light, as you mentioned.
So you know where the mutations are happening, but is it possible that you know where they are happening, but you don't know what will be the result of those mutations?
I would say, for example, in my case, I don't know what would be the results of those mutations. That's why I check my plants also. And sometimes you get something that you don't expect. But with the off targets of CRISPR, for example, at least, as I said, you can select against them. If you see they're there, okay, maybe don't take that plant along. But those off targets, for example, that I saw with some of my experiments, like not all the DNA in your plant has a function. Some of it is what we call junk DNA. So it is a bit of a, like a bad term, because they're also discovering that maybe the junk DNA is not as much junk as they think. But like there's a lot of DNA that doesn't really do much. And if you see it's like in junk DNA, yeah, probably that's not gonna have a big effect on your plant, because it's it doesn't have any function. But you always have to check, of course.
I'm just spitballing here, but if you have DNA and it's classified as junk DNA, but why would it not have evolved to remove junk DNA? Because I would assume if evolution is efficient over long periods of time, that it would have removed those excess parts of DNA.
Well, my first answer to that is that evolution is ongoing as we speak.
Yeah, true. That's a very good answer.
Actually, most organisms have a lot of junk DNA. I think yeast is one of the most efficient, so that it has a high level of genes and way less junk. There's a lot of research on this, so they don't really know why it's there. But one of the hypotheses that they have is that it's maybe good to have junk DNA, because then if the organism is exposed to something that can change the DNA, at least the chance is higher that it happens in a part of the DNA that's not as important.
It's like decoy DNA. So
kind of, yes.
Yeah. Okay. You already talked about CRISPR and how easy it is in use. It has been described even as gene editing you can perform in your kitchen. So how easy is it?
I wouldn't say that you can do it in your kitchen. You don't have at least not the standard kitchen, I would say. Maybe if there's a lab
You haven't
next to the
seen my
kitchen.
kitchen yet, I mean.
Well, maybe indeed you can do it, but I would say feel free. But yeah, no, I don't think you can. It's easy in the sense of, for scientists, if you have the knowledge you need, it's really easy. Can I just get a random person off the street and say, let's do CRISPR, this, you do 1, 2, 3, and that's it? No, not at all. So it's easy in the sense for scientists who are used to doing way harder stuff before, but you need lab skills for sure to do it. And then also what you need even more is, as I said, CRISPR, you can really say which part of the DNA you want to target, but you need to have the knowledge of what you want to target. And that's where your background really comes into play. Otherwise, where do you set your GPS to, if you don't know anything about where you're going?
Well, sometimes you perform random cuts, apparently.
But then if randomness is what you want, I would pick another technique and not CRISPR.
Yeah. Okay.
You're kind of defeating the purpose there.
Yeah, true. CRISPR has been used on people. Why have we used CRISPR on humans, and what was the goal actually?
So, for example, one way CRISPR has been used on humans was somebody who had HIV, and HIV is it's a virus that attacks your white blood cells. But what they also know is that some people in Scandinavia, they're resistant to HIV because in one gene they have a mutation that makes sure that the HIV virus cannot bind to the white blood cells anymore, so they cannot get sick. And so what they did with somebody who had HIV is they took progenitor cells, so it's cells that lead to white blood cells eventually because the body changes them constantly. And there they made a mutation that they saw from the Scandinavians, so that all the white blood cells that would come from that progenitor cell cannot get infected with HIV anymore. I have to say this is all in clinical trial right now. I don't think there's any therapy commercially available right now because CRISPR is quite new and those things, they have to be checked and they take quite a while. But yeah, there was also in China a case where they made a CRISPR or 2 CRISPR babies that was also linked to HIV. That's a scientist who made babies who are already HIV resistant without even having encountered HIV. So it's not, those are 2 different cases. And it's also a very important distinction because the problem with those kiddos is that they have CRISPRed DNA in every cell of their body, meaning also their egg cells or sperm cells, I don't know whether it was a boy or a girl, and they can pass on the mutation in the next generation. And that's a way bigger, I would say, danger because it can spread in the population, as opposed to the person who only has those white blood cells that are CRISPRed is never going to pass on that DNA to anybody else.
And as far as we know, that case in China, is that the only people who are fully CRISPRed in the world right now?
Yeah, that's as you say, as far as we know, yes. Because after that happened, they called together a bunch of people, and internationally that we agreed as a scientific community, like not to do this anymore until we know more about gene editing in humans.
But if I'm not mistaken, that case in China, it also wasn't like officially allowed. They just did it. They didn't ask for permission. Is that correct, right?
Indeed, he did not ask for permission. Now it's all been agreed internationally, we don't. Before that, I think we just all assumed it's common sense that we don't, but you have one person who thinks, let's do it.
Yeah.
And
Okay.
in that sense, yeah, when you say, yeah, CRISPR is easy, if you have somebody with a scientific background who knows how to do it, then it's easy enough also for someone who has done a bad idea that it can happen.
And do you think we have similar or maybe, yeah, let's say similar problems or dangers from biohackers who are using CRISPR?
What are biohackers exactly?
So biohackers are people who try to use tips and tricks or whatever to enhance their own bodies actually at home. And that can go from really simple things like taking supplements, but it can go to injections. And there has been talk, I don't know how much it has been used, of CRISPR, of people using CRISPR at home.
And the question is whether they're a danger to the society.
Yeah, either for themselves or for society.
It's hard to answer this question because I would say for society, probably not, because if they inject it in their body, as I said, they probably won't be making or won't be CRISPRing their sperm cells or their egg cells, and that it gets loose in the the gene pool, so to speak, of humans. And for themselves, yeah, it's hard to say, because the thing is with CRISPR is that you also it works on the DNA, but it also has to reach the DNA of the cells. So it has to be introduced into your cells in order to do CRISPR. And I don't know how well that's been described actually, that we can just inject ourselves with CRISPR, because I guess it would need a coating or something that it can enter the cells. Because if it cannot enter the cells, you're never gonna modify yourself. And even if they find something, yeah, then maybe they're gonna edit one cell out of the many they have. It's hard to predict whether that would have a big influence on them, both in the good and the bad sense, you know.
Yeah.
So it's hard
That's actually, yeah, but that's also a big difference in things that you are saying. And I totally understand, because you have a CRISPR organism when you do it from one cell and it multiplies and it becomes an organism, or you have a fully grown organism and you try to change something in that organism. In that second case, how big can those changes be? If it's already a full-grown organism, can you still have a big effect? Because in my head, it sounds like you're too late to say, let's say it, because it's already a full-grown organism.
Yeah, exactly. Especially for body parts that don't divide anymore. I think this is not gonna be, or you need to have a very efficient delivery system that it enters all your cells. I also don't know much about medicine or CRISPR in medicine to know how far they are. Is there a good coating available to target many cells simultaneously in an organism? And even then, like those biohackers, do they have the knowledge to really target something that's important? Yes or no?
That's
Yeah,
a good question.
it's yeah. I mean, I would never do it, even even if I could probably take a better guess than they do.
Oh,
Yeah,
come on, Evelien, you could grow big muscles or something, or
yeah,
just, I don't know, whatever.
an extra hand or something.
Yeah,
No,
to help in the lab.
yes, and then I can pipe it with 3 hands instead of 2. Maybe I would be faster. Who knows? I also don't know why you would, in their case, why you would use CRISPR. Probably by walking into the sunlight, you have high chances more DNA mutations
Okay,
than
so
injecting CRISPR into your body.
yeah, so let's promote walking in the sun to all biohackers right now. And maybe going a bit more to your expertise, GMOs are a very hot topic. CRISPR plants are actually GMOs, but how do CRISPR plants differ from other GMOs?
First thing that I maybe want to mention is that CRISPR plants are considered GMO in Europe only. So in the USA, Canada, Latin America, even in Australia, and I think also China, they are not considered, they have a different regulation.
What are they called then?
I don't know what they're considered. Well, at least in the USA, they're just considered normal, like any other plants. They don't have any extra, as far as I know. I don't.
Mm-hmm. Yeah.
Yeah. So it's Europe actually who says like CRISPR organisms are the same as GMOs. Don't fully agree with this. So GMOs, genetically modified organisms, actually the term tends to, or at least first when the regulation was made, referred to more transgene plants. So transgenetic plants are plants that in their DNA, they have DNA from another species. For example, you have a tomato plant with a gene from a paprika. And when you go to CRISPR, what you have is that there's actually no foreign DNA that you introduce. What you do is you just edit the DNA of the plant itself. That's then called cisgenesis. So it's a different story, in my opinion, than transgenesis. I agree with transgenesis. You should be careful of how you treat those plants. Also, when it comes to the environment, because if you have a tomato plant with a paprika gene and it can, let's say, pollinate a wild tomato plant, then that also gets it, especially if that has an evolutionary advantage. Maybe you're disturbing nature. And also in case of transgenesis, you can say, okay, that's a change that the paprika gene ends up in a tomato. That's never going to happen spontaneously. It's very clearly artificial. Whereas with CRISPR, the type of mutations you create there, they're actually not different at all from what would spontaneously occur just through evolution, that's DNA mutation also sometimes happen by itself. Doesn't always need a trigger. It's also how evolution happens and what creates genetic variation, and it's why we all look different from each other as humans. And when you see a mutation that CRISPR made, if I don't tell you that this was CRISPRed and you just see the mutation, assuming that you have a background in genetics, you cannot say for sure that this was done by CRISPR, or it happened spontaneously, or it happens because you added a chemical that will induce a mutation. And the fact that you cannot distinguish, and it could just as well have happened spontaneously, is for me a reason to really distinguish them from transgenesis.
Okay, so if I understand correctly, you have cis and transgenesis, is it? Genesis?
Yes.
Yeah.
Genesis, yes.
Okay. And so cis is when you have DNA that is being scrambled of the same species within one plant. And trans is when you insert DNA of a different species into one species.
Exactly.
Okay. And so now in Europe, we say GMOs is all cis, trans, it's all GMO, or not really?
Exactly.
Yeah.
Yes.
And in the States, they say cis is not GMO and trans is.
Yeah, I don't know about the States actually. What the regulation is there. It's for sure more flexible than in Europe related to transgenesis. Because in Europe, for the record, it's allowed. Transgenesis is just you have to do so much paperwork. The plant has to be checked so much that it's incredibly expensive. It takes really long, so that companies tend to not want to do it, or even sometimes just cannot pay to start the whole administrative process. And right now, CRISPR plants are considered the same, so they also have to have the whole check, all the administration done for commercial uses. It can be done always in a research context, but for commercial research, it all has to be checked. And it hampers also a little bit what I've noticed, for example, with funding in Europe. If you say, I'm going to use CRISPR, and you say, okay, I want to apply this for this and this and this, but sometimes they even say like, yeah, but CRISPR is not allowed in Europe. So your whole valorization that you could do is in the trash. So it even sometimes holds back like the more applied research that has a potential commercial application.
Why do you think that is? Do you think that's like because people are just wary of these new technologies, or do you think there are real dangers that have to be considered?
I think neither actually are dangerous, because they're just related to the genetic modification itself or CRISPR itself, because they're just tools. The danger comes from what are you using it for? Because it's a bit like a hammer, a hammer can build stuff. It can also smash somebody's head in. So the technique itself is not inherently dangerous, I would say. But yeah, it's hard for me to predict why politics is making such a big deal out of this. I think it just became a subject for politics to make like their one of the topics that they use, like we're going to be very pro or very con, and some political parties are extremely con against contra.
So in your view, both GMOs in general and CRISPR are safe?
Yes, but with GMOs, I agree that there should be extra caution, mostly related to how there should be systems in place that cannot spread to the environment. But with CRISPR, I don't fully agree that it's really needed to be kept away from the world as much.
So you personally, would you eat GMO or CRISPR food?
Hopefully, yes.
Yeah. So you think that's no problem? I'm not putting in anything in my body that shouldn't be there.
No, because the system that actually does the genetic modification is never in those foods. You're just eating, for example, a tomato that has DNA that has been modified, but the system that did the modification is not there. For example, with CRISPR, we put the CRISPR system in the cell very briefly. It does its work, it goes out, and then the cell grows up with the mutation that we wanted. But the system that did the mutation in the first place is never there. So it's not because you eat a genetically modified food that you're going to get genetically modified yourself.
Yeah, so like the CRISPR and the Cas9 and stuff are already gone. But could you get proteins, for example, in your vegetables that are not that great for you?
When you do this, you know where you're doing the mutation, and you're going to do it somewhere that is not going to affect your food. I mean, why would you make a vegetable that is bad?
No, but I mean, I'm not saying that's intentionally, but scientists are also people. Everyone makes mistakes. Is it possible that there's an adaptation that you haven't seen and that should be dangerous? Now, I want to be clear, that's exactly the same thing for normal breeding. They can also oversee stuff. So I'm not trying to focus on GMO is bad or anything, but are the dangers similar to normal breeding, or is the error of overlooking something, has it bigger detrimental effects than normal breeding, or not really?
The risks there are exactly the same as with normal breeding. And I would even say, because right now the GMO in Europe is regulated as strongly as it is, and it's checked as good as it is, probably you're safer eating a GMO than a regular. It's a joke among scientists, because regular fruit or vegetables that come from normal breeding practices, let's say, they checked way less than the GMOs, because they're turned inside out. So probably it's even safer,
Quite literally, because you're checking the DNA. So
yeah, even that's it.
But you could argue that humanity as a species has lived for a long time without CRISPR. So why do we need it now? Do we need it?
Well, I think, there's a lot of stuff where you can say, okay, we lived so many years without internet, for example. Do we really need it? Yeah, the answer is we could probably survive without CRISPR for sure. But is it gonna be a good tool that can help us with the challenges we're facing in the context of climate change, growing population? Is it gonna be something that is an advantage there to have in your toolbox? I say absolutely. I don't think it's the key to solve everything, not at all. But it's like an extra arrow in your quiver, so just in case that you can use, to help along, both in medicine, for example, but also in agriculture, to make better plants and that are more resistant. And also it can speed up stuff so fast, like also in breeding. It's one of the big advantages of CRISPR, is that you can speed up breeding a lot compared to your normal breeding processes.
And by a lot, you mean like decades?
A decade, in some cases, yes. Because if you have to, let's say you have a mutation in plant A, let's say it's very resistant to a pest. And you have plant B, who has a very, very, very nice yield. And you know from plant A, okay, it's this specific part of the DNA that confers that resistance to pest. If you do normal breeding, you start by crossing the two. But then you have DNA from both a plant that is resistant to pest but has as high of a yield, and you have the DNA from a plant that has a high yield. And then you have to start getting out the rest of the DNA from the first plant. And with CRISPR, what you do is, okay, you have your high yielding plant, you just make the mutation that you know confers resistance to pest in that one, and you have everything in one go. You have a pest resistant plant and with a high yield, and with just regular crossing, that's depending on what the generation time is of your plant, it can really take years.
We talked about cis and trans GMOs, but in classical breeding, they also sometimes use different species to come to one new developed species, let's say. So it's also a form of trans breeding, maybe, or is it seen as safe?
Well, that's actually, making hybrids. It's what it's called, because it happens, for example, I think with wheat, that they make a cross between two species and then you have a new hybrid. That's not considered a GMO. Hybrids are not, it's done quite often even. It's considered a normal breeding practice. So no, those are not under the umbrella term of GMOs. So yeah, the line sometimes is a bit weird. There's a lot of breeding practices, because I think the biggest thing against about let's say GMOs, is it's not natural to do it. But all of our breeding practices, I would argue, are not natural. You have, for example, wheat. That's what we call a hexaploid. Instead of having 2 sets of DNA, one from each parent, it has 6 sets of DNA. So we call it an A, B, and C genome. So compared to its natural or the wild variant, it doesn't even look the same. It's the same with maize. If you compare it to the wild plant, it has a completely different look. You wouldn't even say the same species, actually. So the question you can ask yourself, like, if you say it's not natural, but what is natural? We change also the DNA by specifically selecting for certain traits, and by doing that hundreds, thousands of years in some cases, the DNA is altered also a lot. Okay, you don't do it directly as you do with CRISPR. It's not like you're having, as a scientist, with CRISPR you really have the hand in there, but with regular breeding practices, you change the DNA also, just in a very indirect way.
If we have a CRISPR organism and it escapes into the wild, what are the dangers in that?
Yeah, I think because the mutation could just as well be natural, I don't really see it as a big danger compared to, for example, transgenesis. Whether that mutation is going to survive, that's of course the question, because I can tell you what we want for agriculture is often very different than what a plant needs to survive. That's why we have to pamper them with the very nice soil and irrigation in some cases, and planting and taking the weeds away because they're not competitive. So I think there's no added danger compared to normal breeding practices, for example.
Before we close, do you have a take-home message for our listeners?
Yeah, don't do CRISPR in the kitchen.
Okay, I won't. Okay.
No, but I would say, I think to maybe remember that CRISPR is a tool first and foremost, and that it's a great tool, that has revolutionized multiple scientific fields, among which medicine, but also agriculture. But that it's also something that we should, because I've been very positive here, but I think there should also be some caution related to the technique, and also maybe to be very aware of how we use the technique per se, rather than whether we should use the technique, yes or no.
So like you said, it's a hammer and you can build something with it, but you can also destroy something with it.
Exactly, yes.
This was Apple Finch Pudding. I want to thank Evelien Waegneer for the information. Let's meet again for the next episode of Apple Finch Pudding.